Guys, welcome to chapter 11.3. We are now talking about transduction. So in the last notes, we were talking about the GPCR, or the receptor protein, and now that the message has passed through the membrane, we are talking about how it gets relayed, or how it gets passed from one protein or from one molecule to another until it creates a cellular response. So let's get started. The first thing we're going to talk about is cascades. So if we look here, we have let's call it an epinephrine. We have one molecule that is an epinephrine, and it's going to act on one molecule, and it can potentially act over and over and over and over again on this molecule, which is going to amplify this message. So one epinephrine can act on a single molecule many times. And so this is going to amplify. It's going to create the message. It's going to make the message larger, right? It's going to allow it to be sort of, if you will, a little bit louder. But so what this also does is, so now we have many of these second molecules, but this second molecule could potentially work on more than one type of molecule. So here, you can see how this red receptor protein is going to work on 3 different types of molecules here, and that's going to allow for coordination. What does this mean? Well, it's going to trigger one pathway, but it's going to be working on a different type of molecule, so it's going to trigger another pathway. And it's going to be able to work on another type of molecule, and that'll trigger another pathway. And so these can cause 3 different cellular changes, or 3 different cellular messages, which is going to allow for a larger message to occur. And so it's going to coordinate the change to happen at the same time. And so messages can be amplified. These cascades allow for messages to be amplified, and they also allow for coordination. Both of these are very important. And so the first transduction pathway that we're going to talk about, or the first way that we're going to talk that the message can be passed from one molecule to another, is a phosphorylation cascade. And it's just simply what it sounds like. We're just adding phosphate groups from one kinase to another. So here we have a protein kinase. Remember that a kinase is going to be able to remove a phosphate from an ATP, and it's going to activate. So here we have an activated relay molecule, which we've talked about in the past videos. And that's going to take this inactive kinase, and it's going to activate it. And that active protein kinase is going to do its job, and it's going to add a phosphate group, right, to another inactive protein kinase, and it's going to activate another protein kinase. And so here we have yet another inactive protein, and that kinase is going to take a phosphate group, and it's going to activate that protein. And eventually this is going to cause a cellular response. So we have just a chain of protein kinases just phosphorylating one another, and this is going to cause a cascade. And so about 2% of our genetic code is thought to be instructions for kinases. So these messages are passed from one to another, and they're very important for cell regulation. And so remember that when a message starts, it also has to be able to stop. And so the way that we stop a phosphorylation cascade is through a phosphatase. So there's going to be lots of vocab in this section, so I tried to create this to help you sort of keep the words separate. So our kinases, remember, are going to add phosphate groups. Our phosphatases are going to remove phosphate groups. And so when we have a phosphate, or a kinase, if we were to remove that phosphate group, it's going to deactivate it. And this is going to turn off the signal. This is going to dephosphorylate, and it's going to stop the message. So it's important for us to send messages, but when the message needs to stop, there needs to be a way to stop that message, and that is what the phosphatase does when we are using a phosphorylation cascade. All right, let's now talk about transduction with epinephrine. So remember that we were just looking at a phosphorylation cascade. And so this is going to be a little bit different. This is a different example. And so here we have our transmembrane protein, or our receptor protein. And what we're calling our signaling molecule is a first messenger. This is our first messenger, and it is delivered to the receptor, so we're going to call this epinephrine. And so epinephrine is going to attach to our receptor protein. And another molecule that we're going to be talking about is a second messenger. A second messenger, our example of a second messenger is this cyclic AMP. And it can also be cyclic GMP. These are small ions. These are not proteins, so these are not kinases. These are small non-protein molecules that can diffuse through the membrane because it is water soluble. So remember that an AMP is an adenosine monophosphate. So adenosine monophosphate, one phosphate group. So there isn't a big charge like an ATP with those 3 phosphates. And the C stands for cyclic. And we'll talk about the difference between those 2 molecules. But these are going to be able to diffuse through the membrane because they are small and soluble. So here, those are our first messenger and second messengers. We use these in coordination with other enzymes, so with our kinases. So we have, so we do have proteins like kinases, but we also have these other molecules that are non-proteins. So Earl Sutherland, we talked about him earlier. He was the scientist that discovered that epinephrine can break down glycogen, but it can't do it without other cells, with other living cells in place. And so he found that the binding of epinephrine, or adrenaline, to a liver cell causes an increase in cyclic AMP in the cell. And so what happens here, what he's discovered is this pathway. Let me make this a little bit bigger. I'm going to cover up that. And we have our first messenger, our adrenaline, or our epinephrine. It's going to bind to our GPCR, and that's going to allow our G protein to interact with our enzyme. And in this case, the enzyme is adenylyl cyclase. And adenylyl cyclase is then going to clip off 2 phosphate groups to create cyclic AMP. So it's going to take an ATP, and it's going to clip off 2 phosphates to make it a CAMP. So let's look at our reaction, our pathway here. The ATP, right here, we have an ATP, and adenylyl cyclase is going to clip off 2 phosphate groups, and it's going to make cyclic AMP. And cyclic AMP is an adenosine monophosphate, but it has this cyclic, it has this little circular shape on it. And it is going to be able to, this is our second messenger, and it's going to interact with a protein kinase, and it's going to which is going to allow the cell for the cellular response, which is to break down glycogen into glucose. So the cyclic AMP is going to make the message happen. It's going to send that message to protein kinase A, and the protein kinase is going to tell the cell to break down glycogen into glucose. At some point, that message needs to stop. And so there's a phosphodiesterase. Remember that we need to start messages, but we need to be able to inhibit them as well. And so what phosphodiesterase does is it's going to clip that cyclic part, and it's just going to make it an AMP. And this is going to be, or an adenosine monophosphate, and that's going to inhibit, that's going to stop the message. And so adenylyl cyclase activates, and the phosphodiesterase deactivates. So to understand the pathway, the epinephrine pathway, there's a little video here, and I'll put that in the discussion or the description. And I recommend watching it because it'll just help you wrap your head around this. And so here, we're going to talk about a couple of different examples of diseases that are caused by problems with the transduction pathways. The first disease we're going to talk about is cholera. What is it that causes cholera? Well, it has to do with this transduction pathway. What happens is that cholera is a bacteria that causes diarrhea. And how does this happen? So we have a bacteria that causes a biofilm, which we talked about. The bacteria are in a biofilm. Bacteria are in a happy spot, or in a healthy spot, and they proliferate. They're going to produce many more bacteria, and they send out this biofilm that's saying, multiply, 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 and create specific enzymes or create specific proteins. And so this cholera creates this biofilm on the lining of the small intestine. So if you get it, then it's the happy place, the healthy spot for this bacteria is it's gonna land in the intestines, and it produces that biofilm. And what it does is it modifies the G protein. So remember, that's the little guy between the receptor and the adenylyl cyclase, and it's going to it's going to turn it on. It's going to not allow this G protein to deactivate. And so the message is going to be continuing to send, and it won't stop sending. And the message that it's sending is to allow salt into the cell, into the cells of, or into the small intestines. And so if you put salt into the small intestines, we know that that's going to pull more water in too. Water is going to rush into the intestines, and that's going to cause diarrhea. Too much water in the stool is going to cause diarrhea, and eventually it's going to remove enough water out of the body that it can be devastating. It can people die from cholera all the time because they dehydrate. And so there's a video here on cholera, but that is how this interruption of the transduction pathway can cause a disease. Next category or topic is Viagra. So instead of a disease, we're looking at a cure. So the pathway for Viagra is going to include cyclic GMP, which is guanosine monophosphate, just like AMP. And once again, it's similar to ATP. And using this GMP, doctors have created Viagra. So we know that Viagra is going to dilate the blood vessels throughout the whole body. And the increase in blood in the penis is going to allow for an erection. And so when nitric oxide is released by neighboring cells, the cyclic GMP is the second messenger. So here's our cyclic GMP. It is our second messenger that signals muscles in the arteries to relax. But what Viagra does is it inhibits the hydrolysis of cyclic GMP, meaning that the second messenger stays on. It doesn't get inhibited. It doesn't get turned off. And so therefore, the arteries continue to stay dilated and blood flow continues, and so the erection continues as well. And so here, our final transduction pathway that we're going to talk about, and this is calcium, IP3, and DAG. So remember that calcium is a more abundant second messenger than cyclic AMP. It's used in cellular responses like muscle contractions, exocytosis, and cell division. And so we know from talking about nerves that there's always some calcium inside of a cell, but there's always a lot more on the outside of a cell, in the bloodstream and in the extracellular fluid. There's also lots of calcium in the endoplasmic reticulum, in the chloroplasts, and in the mitochondria. So there's lots of calcium found in specific organelles. And so we're going to, this process is going to open up the membrane to allow some of that calcium into the cytoplasm. And so remember that there's not usually much calcium in the cytoplasm. So if we increase it by a little bit, even if we increase it by a little bit, it's going to have a large change on the percent. It's going to create the percent change is great. And so we are going to, there's going to be 2 second messengers involved in this process, IP3 and DAG. So let's look at the pathway here. So remember that the goal of this is to open up a membrane to allow calcium out of, in this case, it's going to be endoplasmic reticulum, to allow the calcium out of the endoplasmic reticulum and into the cytosol, where that calcium can work on other proteins. So here, let's look at let's start at the beginning. Here's our first messenger. It's going to attach to our signaling protein, or our receptor protein. It's going to activate our G protein. And instead of an adenylyl cyclase, we have a phospholipase here. And the phospholipase is going to cleave a phospholipid from the membrane. And it's called a PIP2. It converts it, it takes the PIP2 and converts it into 2 different things, a DAG and an IP3. So the DAG remains in the membrane, is used for other things. We're going to follow our IP3. So this PIP2 is going to be cleaved. The DAG stays here, and we're following the IP3. The IP3 is going to be used it's going to attach onto a calcium-gated channel that's found in the endoplasmic reticulum. The endoplasmic reticulum, remember we said there's calcium in there, and so this is going to open up that gate and allow the calcium to come out of the endoplasmic reticulum and enter into the cytoplasm. So the goal of this IP3 was to act as a signaling molecule onto this calcium channel. It's going to open the channel and allow calcium into the cytoplasm. And so here, this calcium is going to work on other various proteins. And so it could work on a number of different proteins, creating a number of different messages, or cellular messages. And so this is one way that we're going to coordinate or to organize. So there are many, many steps in these processes. I would strongly recommend that you draw these things out. Start with the phosphorylase, or the phosphorylation cascade. Wrap your head around that. I would then draw out the transduction with epinephrine, and then eventually I would draw out the IP3, calcium IP3 and DAG. If you have any questions, please feel free to reach out.